right, let's go through paper 4, which is the extended theory paper for biology. This is from Cambridge IGCSE, Cambridge International Examinations, CIE. This is May, June 2016, variant 2, so the code is 0610/42. All right, let's get started.
Let's get started.
Question 1. All mammals have a double circulatory system. Figure 1.1 shows part of the human double circulatory system. Okay, so let's figure out what's happening in this diagram. This is figure 1.1. Okay, this is a bit of a confusing picture of the heart, but if you look at it, the blood is flowing from the body in through this vessel into chamber A. Then it goes down into chamber D through the atrioventricular valves. Then this chamber D contracts and it goes up to each lung. Okay. Then in the lungs it becomes oxygenated and blood enters, re-enters, just going behind that vessel into B, and this side also enters into B, and that's the same chamber. The B is the same chamber, so then they both go down into C. Then this chamber contracts and pushes the oxygenated blood out all the way to everywhere else in the body other than the lungs. So that is blood flow through the heart and through the lungs. And so now if you follow the red arrow here from the aorta, keep going down and go down through this vessel here. It will go to one side, then the other side will go to one kidney, then it will do what it needs to do its gas exchange, and then come back out of the kidneys down here. And then go up, back in to chamber A. All right, so that's the full cycle. A, name the muscular wall that separates the left and right sides of the human heart. Okay, the muscular wall is the septum. B1, describe what is meant by the term double circulation. Okay, so double circulation, what it means is that the blood goes through the heart twice in one cycle. So blood flows through the heart twice for one complete cycle. One loop is to the lungs and one loop is to the rest of the body. So pretty much you don't have to put both sentences down, choose one or the other, but if you do the second sentence, make sure you say one loop to the lungs and one loop to the rest of the body in one complete cycle. So the key thing is one complete cycle, 2 loops through the heart. 2, state one advantage of a double circulation. There's lots of advantages, actually. One of them is that by having a double circulation, you can have a different pressure of blood to the lungs as compared to the rest of the body. So it allows each loop to have a different blood pressure. Another thing that it does is it means that the body can actually have a larger blood pressure in total. And so that will increase the flow rate of the blood than if it were single circulation. It also means that the oxygenated and deoxygenated blood won't mix. Another thing that it does is it allows animals to have a higher metabolic rate. And it also allows animals to be much larger, much taller. Okay, fish have a single circulation and they have maximum sizes that are a lot smaller. C. Table 1.1 describes some of the structures of the human circulatory system shown in figure 1.1. Complete the table and one row has been done for you. So we have the name of the structure and we need to figure out what letter it is on figure 1.1. So first of all, let's figure out the name of the structure. Okay, the heart chamber with the thickest muscular wall. Well, that is the heart chamber that needs to send blood pumping to the entire rest of the body. That would be the left ventricle. Okay, the blood vessel that carries oxygenated blood to the heart, that is the pulmonary vein. Pulmonary, because it's just become oxygenated by the lungs. So pulmonary means lungs, and vein, because it's going towards the heart. The blood vessel that carries oxygenated blood away from the heart, Okay, away means artery, but this one isn't an artery. It doesn't say artery in the name. It's the aorta. It's the largest artery in the body because it needs to take all of the blood to push it to the entire body. And the blood vessel that carries blood away from the kidneys. So kidney means renal. And if it's going away from the kidneys, it's going towards the heart. So that is vein. Okay, so we need to figure out where the left ventricle, the pulmonary vein, the aorta, and the renal vein are. The vena cava, which is the blood vessel with the largest lumen, or space in the middle, has already been done for you. That's letter N. Okay, so the heart chamber with the thickest muscular wall, as we said, that's the left ventricle because it needs to send blood to the entire body. If we divide the heart, this is how we divide it into two, the left and the right. I know it's at an angle, but that's how it is in this diagram. Okay, so this is on the left side of the picture, so it's the right side of the person. And this is on the right side of the picture, so it's the left side of the person. The ventricles are on the bottom, so C is the left ventricle. Okay, the blood vessel that carries oxygenated blood to the heart. Oxygenated blood to the heart, that would be K. That, as we said, that was the pulmonary vein. The blood vessel that carries oxygenated blood away from the heart, that's this one here. And that is, where's the letter to that? That is the aorta. Ah, there it is. It's P. That's the aorta. Okay. And the blood vessel that carries blood away from the kidneys, that's this one down here. That is M. That is the renal vein. All right. So we have left ventricle is C, the pulmonary vein is K, the aorta was P, and the renal vein was M. All right. If you make a mistake on one of these, don't try and correct it over top. Don't say that, oh, that should be D. It's not D, but it's C. Instead of writing over top of it, make sure you cross it out, maybe even scribble it out fully or just one line, doesn't matter, and write it next to it. Because if you write it on top and there's any sort of confusion, if you say that's a B or a P, then they go, oh, which one did they actually mean? And so you can lose a mark because I don't know which one you meant. All right, that's it. D, describe how blood is transported from the vena cava to the lungs. You may use the letters on figure 1.1 in your description. You don't have to use the letters in figure 1.1. I would actually say in this particular question, I'd say it'd be better to write in the structure, because if you use the wrong letter, if you say what it is and then you use the wrong letter, well, you'll get it wrong. Okay, to do this, I'm going to make my simplified diagram that I always do on things like this. Okay, so whenever I'm doing labeling of the heart, I always like to do this nice simple diagram. Okay, so you draw a heart. I know they say that the heart doesn't look like an actual heart, but it kind of does. Okay, and you divide it into four quarters. Okay, so this is the left hand side of the diagram, so it's the right hand side of the heart. This is the right hand side of the diagram, so this is the left hand side of the heart. These are the atria, or atrium if you're talking about one, and these are the ventricles. Okay, so we have a vessel here, a long vessel here, a long vessel here, and a vessel here. Okay, we have, these are two valves here. These are the atrioventricular valves. And we have some vessels here. These are the semilunar valves. Semilunar valves. All right, now let's look at the blood flow. All right, so deoxygenated blood goes in from the body down into the right ventricle. Then it gets pumped up towards the lungs. Okay, in the lungs it becomes oxygenated. Okay, and then from the lungs it comes in, goes down into the left atrium and left ventricle, and it goes back up through the aorta to the entire body. Okay, so that's what happens. That's as simple as you can make it. Okay, so let's now put that into words. So the blood enters the heart through the vena cava, goes into the right atrium, then the atrium contracts. Blood flows through the atrioventricular valve to the right ventricle. Then the ventricle contracts and pushes the blood into the pulmonary artery. The blood passes through the semilunar valves in the pulmonary artery, and goes towards the lungs. Okay? Now, if you look at this, you may use the letters on figure 1.1 in your description. Only do that if you are 100% sure that you won't make some sort of crossover mistake. It's very easy to just write in the wrong letter. If you already know the names, just write in the names. E1. Doctors recommend that a healthy diet can reduce the risk of coronary heart disease. Give one other lifestyle improvement patients can make that can reduce the risk of coronary heart disease. Keyword there is one. There's more than one you can put down. Only put down one. And another one is other. Do not mention anything about diet because it says so in the question. Okay, so another thing that can reduce the risk of coronary heart disease, more exercise. Okay, you can write that in a sentence. You don't really have to, though. Also, you could say, stop smoking. If you're smoking, stop smoking, because that will reduce the risk of coronary heart disease and reduce stress levels. All of those things will give you a good mark. 2. Sometimes surgery is required to treat coronary heart disease. Describe one named example of surgery that can treat coronary heart disease. Okay, one thing you could say is you could discuss stents. So stents can treat coronary heart disease. How? So a small mesh tube is inserted into the artery to open and support narrow or weak arteries. Another thing you could say is balloon angioplasty. So a tube or a catheter is just a tube, with a balloon attached is inserted into the artery. The balloon inflates to widen the artery. Okay, another thing that can be done is bypass surgery. Okay, and in bypass surgery, there's another blood vessel it has to be an artery. You can't put a vein onto an artery and expect it to act like an artery. So another artery is joined to the artery to allow the blood to flow along a new route. Okay, so either of those three will work. Question 2. Figure 2.1 is an electron micrograph showing the bacteria Vibrio cholerae. Okay, that's cholera. A1. Bacteria are prokaryotes. State 2 distinguishing features of all prokaryotes. Keyword there is all, and obviously only 2. Okay, lots of things you could put down here. 1, you could say that they have no true nucleus. So no true nucleus, no nuclear membrane. That's what prokaryotes mean, before a kernel, before the nucleus. Okay, they also have no membrane-bound organelles. So no membrane-bound organelles. And remember, when you're doing this, ribosomes are not membrane-bound organelles. They do still have ribosomes. They're also single-celled, and they have a loop of DNA and plasmids. Okay, they do have a cell wall, and it's a peptidoglycan cell wall. It's not a cellulose cell wall as it is in plants. And they reproduce by binary fission. Okay, so the ribosomes, they do have ribosomes. They're smaller than eukaryotic ribosomes. They're 70S. Okay, that's just a unit they use to measure it. Now, one thing you can't say that is very tempting from this picture, the flagellum is a very obvious feature of these bacteria. Well, flagella aren't in all prokaryotes. They're in some prokaryotes. And capsules. Many prokaryotes have capsules, but many don't. Pili. Many have them, many don't. Cilia, same thing. So you can only choose things that are in all prokaryotes. 2. The bacteria shown in figure 2.1 each have a flagellum. Suggest the function of the flagellum in bacteria. The function of the flagellum in bacteria is exactly the same as the function of the flagellum in sperm. It is to move the bacteria. The function of the flagellum is to allow movement. Okay. B. V. cholerae is a pathogen that causes cholera. Vaccination is used to control the spread of cholera during an outbreak. Explain how vaccination can control the spread of diseases. Okay, so first of all, what is a vaccine? And a vaccine is a harmless form. It can be attenuated, so it can be a harmless form, or it can be a dead form of the pathogen. And this is either injected or swallowed. More often it's injected. So it's a harmless, attenuated, or a dead form of the pathogen, and it's injected or swallowed. Sometimes it's just the antigen. They're not injecting antibodies, they're injecting the antigen so that the immune system can react. So the antigen, what is it? It's specific to the pathogen that it's being vaccinated against. The antigen is specific to the pathogen being vaccinated against. Okay, and what happens once it's injected? The lymphocytes respond to the antigen by making antibodies. So lymphocytes respond to antigens by making antibodies. And over time, some of those lymphocytes will become memory cells, and they'll be able to make those specific antibodies very quickly if there is another instance of that pathogen in the body. So memory cells that are able to make more of that specific antibody remain in the body for a long time, and so this means that you can have a very rapid response next time. So next time the body is exposed to the pathogen, it can have a very rapid response by producing antibodies quickly. Okay, other things you can mention, you could say this is active immunity. And you could also say about if most people are vaccinated, that creates herd immunity. So some people, even if they're vaccinated, are not going to have immunity to the pathogen, but if you get the majority of people being vaccinated, then you're unlikely to have an outbreak because you're not able to spread the pathogen. Okay, and there's other valid points you could put down. You could talk about details of active immunity, and you could talk about how smallpox has gone extinct due to vaccinations. C. Many years ago, scientists discovered that V. cholerae secretes a toxin. Figure 2.2 shows the result of an experiment to measure the flow of chloride ions out of human cells with and without the toxin. Okay, so if there is no toxin present, then chloride ions slowly go out of the cell. But if there is a toxin, it means that chloride ions quickly go out of the cell, and you never get them quite back in the same way. All right, so here we have arbitrary units, and we have time in seconds. All right, if you notice on this graph, there's two lines, and one of them is drawn with an X, one of them's a dot with a circle, and those are the only ways that they really like you to draw points on your exams. 1. Calculate the difference in flow of chloride ions between the cells with the toxin and cells without the toxin at 50 seconds. Show your working and state the units in your answer. Okay, it does say state the units in your answer. Okay, make sure you read that. All right, so you want to calculate the difference. That means that's the amount with the toxin minus without toxin. Okay, flow of chloride ions with the toxin, flow of chloride ions without the toxin, and we're looking at 50 seconds. All right, so 50 seconds is here. All right, so each one of these marks, if we have 2 arbitrary units divided by 5 squares, so that means each square is 0.4 arbitrary units per square. Okay, so every time you go up one little square, you're going up 0.44 units. All right, so if we look at this, here for without the toxin, it's at 0.4. Let's draw the line to show that. Okay, and with the toxin, it's at 12. All right. As we said, the units are arbitrary units. Okay, so we have with toxin, that is 12 arbitrary units. Without the toxin, 0.4 arbitrary units. That means that the difference is 11.6 arbitrary units. So 11.6 arbitrary units. You've included the units in your answer. 2. Use the data in figure 2.2 to describe the effect of the toxin on the flow of chloride ions out of the cell. When it says use the data in figure 2.2, 1 mark will be for a data quote. And remember, you're not just quoting 2 random points. You want to quote 2 important points, a peak, or when it gets back to a stable level or something like that. You want to quote something useful, not just something that's in the middle of not really much happening. Another key point about this question is that you're describing the effect of the toxin on the flow of chloride ions out of the cell. You're not comparing the two, and you're not even mentioning anything about the normal. Okay, you're just describing what happens when the toxin is present. Okay, so what happens at first? At first, there's a very large and rapid increase in the flow of chloride ions. Okay, so a very quick increase. All right, and then it peaks at 50 seconds, and it stays fairly constant, gets down to about here, and it starts staying fairly constant. So that's at about 125 seconds. Okay, and all the way to 250 seconds. Okay, so it just says describe. It doesn't say anything about explaining. So first of all, there's a large and rapid increase in the flow of chloride ions. And then, so it peaks at 50 seconds with 12 arbitrary units and decreases to around 5 arbitrary units by 125 seconds. So it stays fairly constant between 125 seconds to 250 seconds at about 4.4 arbitrary units to 5 arbitrary units. So every time you quote a data point, you always put the units. All right. Or else it doesn't really count. 3. Chloride ions cannot move out of cells by simple diffusion. Suggest and describe how chloride ions could move out of cells. Suggest means you don't necessarily know 100%, but you're using the information you're given to give a good answer. Okay, so if it can't move by simple diffusion, that means it has to move against the concentration gradient. And whenever it moves against the concentration gradient, it needs energy, and that's active transport. All right. So chloride ions can move out of the cells by active transport through proteins and cell membranes. Okay, active transport requires energy. So this requires energy, or you could say ATP, from respiration. Okay, and whenever you mention anything about diffusion or active transport or anything like that, you should say whether it goes against or down the concentration gradient. So it moves the chloride ions against the concentration gradient from low to high. So it moves the chloride ions against the concentration gradient from low concentration to high concentration. D. The loss of chloride ions from cells causes diarrhea and dehydration in patients with cholera. 1. State which organ in the alimentary canal is affected by the cholera toxin. Okay, so if it causes diarrhea, it will be the small intestine affected. So the small intestine. And 2, describe the treatment for cholera. So this is two marks, but there's a few things you can put down. It's called oral rehydration therapy, and it involves a drink that is a mixture of sugar and salt to help replace the water lost. That water in the drink has to be clean water, boiled, or another method of being sanitized, so that you don't get sick again. And another thing you can do is you can use antibiotics to kill bacteria. So both of them together. So first of all, you treat the diarrhea, and you can kill the bacteria. So oral rehydration therapy is a drink of a mixture of salt and sugar to help replace lost water. And the water in the drink must be clean, because generally when you get cholera, you get it from dirty water. So that's an important point. And you can also use antibiotics to kill the bacteria. All right, let's move on to the next question. Question 3. Anthocyanin is a red pigment found in carnation flowers. Some carnation plants have a gene for making anthocyanin. A1. A flower grower bred red carnations. Describe how growers selectively breed plants. All right, so basically what they do is they crossbreed the parent plants that have the desired characteristics, and then they choose the offspring that have the desired characteristics, and they crossbreed these with the original variety, or they can do self-pollination, or they can breed them with the other offspring. And this happens over many, many generations. So growers crossbreed parent plants that have the desired characteristic. They then choose the offspring that have the desired features and cross them with either the original variety, they can self-pollinate them, or they cross them with each other. You don't have to write down all those. You can write down 1 or 2 or 3. That's okay. And they do this many times. So they do this for many generations of crossing and selecting. Okay. You can also put in any other details. You can describe how they bag the flowers, how they transfer the pollen with paintbrush, how they collect the seeds, but those are the main points. 2. Explain the disadvantages of using sexual reproduction to breed red carnations. Well, to start with, it needs 2 parents. Asexual reproduction only needs 1. It requires 2 parents. Another problem with sexual reproduction is that sexual reproduction results in genetically different offspring. So that means they may not all produce red carnations. There may be other colors in there. There is variation in the offspring, so they might not all be red. Also, it takes a lot of time. It takes a lot of time. Time for many generations, and also time to take a little paintbrush and paint the pollen from one plant onto the stigma of another plant. Okay, and you can do any other valid point as well. You can mention anything about harvesting seeds, finding pollinators. Those can be quite tricky, how there can be a limited number of seeds, and how a lot of the pollen is wasted. B. Meiosis is necessary for sexual reproduction of carnation plants. Define the term meiosis, okay? So, mitosis makes mitose, and meiosis makes everything except mitose, so the sperm and the egg. Obviously in plants it's pollen and egg. All right, that's how I remember those 2. That's not your answer. That's just a quick way to remember it. Okay, so meiosis, it's known as a reduction division because it takes the chromosome number and halves it. So it goes from diploid to haploid. So meiosis is a reduction division, so the chromosome number is halved. Cells go from diploid to haploid. All right, and what does this result in? Genetically different offspring. Oh, not offspring, sorry. Genetically different gametes. Okay, it's sexual reproduction, they need to be different, and this doesn't result in the offspring, it results in the gametes, the sperm and the egg, or the pollen in the egg. It results in genetically different gametes, not offspring. We're not there yet. You have to combine the sperm and the egg before you can get offspring. All right. C. Carnation plants show co-dominance for the anthocyanin gene. There are 2 alleles. We have the F capital A allele for anthocyanin for the pigment, which means it has red flowers, and F capital N allele for no anthocyanin pigment, and that creates white flowers. If you notice both of these alleles, it's capital. It's a capital letter, which means that they're both dominant, as it says. When you write them, you tend to write the first in the alphabet first. So it says 1, state the genotype of a carnation plant that is heterozygous, which means it's different for this gene. So we have FA, FN, and that's it. 2, describe the phenotype of a heterozygous carnation plant for this gene. Phenotype means what is seen. What do you see? You don't see a red, you don't see a white, you see a mixture of half red, half white, and that would be pink. It will have pink flowers. Good. 3, the breeder crossed a FAFN carnation plant with a FAFA carnation plant, a heterozygous with a homozygous. Predict using a genetic diagram the proportion of pure breeding carnation plants in the offspring. Okay, it even says what to put into the genetic diagram. Okay, so the gametes, you just divide those up. We have FA, FN. And this says FA, FA. You just divide them up. Our Punnett square, well, we have FA, FN, FA, FA. Then we draw in some lines, and then we just fill in the cross. So we have A, FA, FA, FN, FA, 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 FN. There we go. So now we need to figure out what the offspring genotypes are. So genotypes, what do the genes look like? So we have FA, FA, and FA, FN. Those are the two types of genotypes we have. Offspring phenotypes. So we look, if we, the A, as we saw up here, the A means red flower. So if you have two A's, they're going to be red. So red and, well, a red and a white is a pink, as we've already said. And proportion of pure breeding carnation plants, it can be, you can say it's, so here are the pure breeding ones. The pure breeding are the homozygous. Okay, so proportion, we can say it's 50%, or we can say it's 1 to 1, or we can say it's 0.5, or we can say it's half. They're all different ways of saying proportion, and they're all correct. Just write one. Okay, so pure breeding means that they're homozygous. Question 4. Some students investigated osmosis in raw potato sticks. A. Define the term osmosis. So, another definition you need to know. Osmosis, the movement of water molecules from high water potential to low water potential, down the water potential gradient, and across a partially permeable membrane. Those are very important things. You just need to remember each part of it. So osmosis is the movement of water molecules. That's 1. From high water potential to low water potential. That's 2. You can also say down the water potential gradient. That's another way of saying 2. You don't have to put both of them. You can if you want. You don't have to. Across a partially permeable membrane. That's 3. Okay. Now, remember, it's water potential, not water concentration. Concentration means the stuff that's dissolved in the water. We're talking about the water, not the stuff dissolved in the water. The opposite of concentration. Okay. B. The students measured the mass of 4 potato sticks using an electronic balance. Figure 4.1 shows an electronic balance. All right, that's an electronic balance. The students left each potato stick in one of 4 different liquids for 5 hours. One was distilled water, one was 0.1 mole per decimeter cubed sodium chloride, one was 0.5 mole per decimeter cubed sodium chloride, and one was 1 mole per decimeter cubed sodium chloride. After 5 hours, they measured the mass again and calculated the change in mass. Okay, so distilled water, that is a high water potential outside the potato stick. 1.0 is the lowest water potential outside the potato stick. Okay, and it just goes in that order. All right. 1. Predict which of the liquids would cause the largest decrease in mass of a potato stick. Okay, for decrease in mass, you'd have to have your potato stick. You have to have the water moving out. The water is going to move into the direction where there is more salt and less water. Okay, so which one of these basically has most salt? Okay, the one with the most salt is this one, 1.0 mole per decimeter cubed sodium chloride. So that's 1.0 mole per decimeters cubed sodium chloride solution. All right. 2. The students dried the potato sticks with paper towels before putting them on the electronic balance. Suggest why. Well, the reason why is because I don't want to weigh the water that's on the outside of the potato stick. To remove water on the surface of the potato stick so that only the mass of the potato stick was measured. This is not the dry mass. The dry mass is then drying it out. It's just the mass of the potato stick. C. After the experiment, the students noticed that the potato stick with the lowest mass was soft and floppy. Explain why the potato stick had become soft and floppy. Well, the reason why is because cells in the floppy stick have lost the water by osmosis. The cells in the floppy stick have lost the water by osmosis. And then you can describe osmosis because it's saying explain. So it's pretty much exactly what you've just explained. So the water has moved from high water potential to low water potential. Then you put in some nice key words that they like to see. The cells were plasmolyzed and have become flaccid as they have lost turgor pressure. Remember, plasmolyzed means that basically the water inside the cell has left. They've become flaccid. Flaccid means basically floppy. And they have lost turgor pressure. And the reason why is turgor pressure is basically the water pressure inside the cell. So there's not enough pressure of water pushing on the cell walls to keep it firm. So that's pretty much what turgor pressure means. D. The students followed the same experimental procedure with boiled potato sticks and found no overall change in mass in any of the solutions. Suggest why the mass of the boiled potato sticks remained the same. Well, when you boil potato sticks, you basically denature any proteins that are in membranes and you destroy the membranes. And remember, the definition of osmosis is it's across a partially permeable membrane, so it won't occur. So when the potatoes were boiled, the proteins denatured and the cell membranes were destroyed. No osmosis can occur if there's no cell membrane. Cell walls have nothing to do with osmosis. They're just basically a mesh on the outside to make it so that the cell doesn't burst. 5. Hormones are secreted by glands or made artificially by drug companies. A1. Name the gland that secretes testosterone. And the gland is the testes. The testes are actually glands. They secrete testosterone. 2. State why testosterone can improve sporting performance. Well, testosterone has a few effects that can really help with sporting performance. It can help with muscle strength, improve recovery of muscles when they've been damaged. It increases a competitive drive, and it also increases bone density and bone mass. Testosterone increases muscle mass and strength. It also helps improve recovery of damaged muscles, increases competitive drive, and increases bone density and mass. Okay, for the 1-mark question, you don't need all that information. That's all the information you can put down, but you don't need to put down all of that. Only put down things you know for sure are true. 3. Describe the role of progesterone in the menstrual cycle. Okay, progesterone maintains the lining of the uterus, and that's known as the endometrium. Progesterone maintains the lining of the uterus, known as the endometrium, and it also inhibits FSH and LH from being released. So it inhibits FSH and LH from being released, and that's why progesterone is generally produced after ovulation. 4. Synthetic progesterone is found in oral contraceptives. Name one other hormone that can be found in oral contraceptives, and that is estrogen. Estrogen, remember? You have that sneaky little O at the front of estrogen. Water entering 2 sewage works, A and B, was tested for the presence of 4 hormones. The testing was repeated on water that left the sewage works to flow into Lake A and Lake B. The results of the tests on the water samples are shown in Table 5.1. Okay. So we have the sewage work A and B, before sewage treatment, after sewage treatment, both of them. And in sewage work A, they did ozone and chlorine, and in B, they just did chlorine, but with no ozone. All right. The hormones they were looking for were estrogen, synthetic progesterone, natural progesterone, and testosterone. Okay, so let's look at the questions. B. Explain which water sample was most polluted with hormones before sewage treatment. Use data from Table 5.1 to support your answer. Okay, this one it even says use data from the table, so you must use data. So we're looking at before treatment. If we look at this, estrogen, not measurable at A and has a tiny amount at B. But synthetic progesterone, almost double at A. Natural progesterone, 2.5 compared to 2.4, slightly more. Testosterone, a lot more, 15.6 compared to 2.7. Okay, so I would say that A is more polluted with hormones than B. You could say B has more estrogen, but everything else there's more of the hormone at A than there is at B. Although natural progesterone, I would say that that is practically exactly the same in both of them. Certainly the progesterone and testosterone, big differences. Okay. So A is most polluted before treatment as it has the greatest overall concentration of hormones, specifically the testosterone. So all the hormones are at a higher concentration except estrogen, and there is a very similar level of natural progesterone in each. All right. And for each of these points, you could say, we're saying A is most polluted. You could say B is least polluted instead. So you could do it from the perspective of A being most polluted or B being less polluted. So testosterone levels in A were 15.6 nanograms per decimeter cubed, and they were only 2.7 nanograms per decimeter cubed in B. The reason why I chose the testosterone to quote as a data quote is because it's the one that has the biggest difference, makes the most effect on your claim that A is more polluted. So that's 3 good points. You could say these 3 points, or you can't say both. You can't say A and B. Or you could say that B is more polluted because there's more estrogen and more types of hormones. So if you look at this, so estrogen goes from not measurable to 0.1 nanograms per decimeter cubed. That doesn't sound like very much. And like B has 4 types of hormones and like A has 3. So here are 2 points that you could, so more estrogen and more types of hormones. And the other thing is a data quote as well, a valid data point. So probably that it's not measurable at A, and there it's 0.1 nanograms per decimeter cubed at B. You have to put the units in, or else they don't count it as a data quote. Data includes units. C. One reason for treating sewage is to reduce the concentration of hormones in the environment. 1. Chlorine was used in the sewage treatment of both lakes. Describe the effect that chlorine had on the hormone concentrations in the water. Use data from table 5.1 to support your answer. All right. So now we're basically looking at the differences between before and after treatment. And remember, this Lake A includes ozone, and Lake B did not include ozone. So if we look at estrogen before and after treatment, well, obviously sewage work at A made no difference because it was not measurable in either one, but in B it reduced it a bit. And if we look at progesterone, it decreased it when there was ozone, but it didn't really decrease it without ozone. Either there was very little effect, or it went up. It could just be a measuring difficulty with measuring it. Okay, with natural progesterone, it went up actually for both instances. So with natural progesterone, the treatment increased the levels for both of them. But testosterone, with ozone and chlorine, it dramatically reduced the levels, but without the ozone, it increased the levels. So we can mention those in our answer. So the levels of estrogen in B decreased slightly. You can't tell in A because it was not measurable in before or after. So progesterone levels increased slightly in both A and B. So there was little effect on estrogen, progesterone, and testosterone without the ozone. So the chlorine with ozone caused a decrease in testosterone and synthetic progesterone and an increase in natural progesterone. Okay, this is obviously a lot more than 2 marks. You don't actually have to mention what happens to every single thing. You could say the levels of estrogen in B decreased slightly, progesterone levels increased slightly in both A and B, and that would be okay because that's 2. All right, so you don't have to mention every single thing. You can say there was little effect on estrogen without the ozone. You don't have to mention everything. Okay. 2, state the main purpose of chlorine in sewage treatment. Well, the main purpose of chlorine is to kill the bacteria or pathogens and to make the water safe. To make the water safe by killing the harmful pathogens. All right. D, describe the negative consequences of letting untreated sewage flow into lake ecosystems. Well, one of the big negative consequences is eutrophication. It can cause eutrophication. And eutrophication is basically where you put sewage or manure from farmers' fields, where that drains off into the natural lake ecosystems, and it fertilizes the algae. The algae grows on the surface of the water and it blocks the sunlight. All right, and when it blocks the sunlight, other plants in the water end up dying. And when the other plants die, then bacteria decompose those plants. And those bacteria do aerobic respiration. They use up the oxygen, produce carbon dioxide. The fish die because they don't have enough oxygen. And carbon dioxide is an acidic gas. So the fish die because it's become more acidic. It makes the lakes all ugly and smelly. It disrupts food chains. And that's basically eutrophication. So let's try and describe that here. So algae grow on the surface of the lake and block light below. This makes the plants below die. So the dead plants are decayed by bacteria that do aerobic respiration. This decreases the oxygen concentration and increases the acidity of the water because of the carbon dioxide produced. Aquatic animals such as fish die because of the lack of oxygen and the increased acidity. When food chains are disrupted and the lake looks and smells bad. Okay, that's one big thing that can happen with untreated sewage flowing into lake ecosystems. But that's not the only thing. Another thing is that untreated sewage can contain heavy metals, which are not very good for aquatic animals. So untreated sewage can contain heavy metals, which are toxic for aquatic animals. And in here you can put in something about bioaccumulation, if you would like. And bioaccumulation or biomagnification is basically where you have your food chain, and let's say here's your pyramid of numbers. It's not always a pyramid for pyramid of numbers. So you have your eagle at the top and you have plankton at the bottom, and maybe you have some fish in the middle. Okay, very simple. Okay, lots and lots of plankton don't have much heavy metals in their system, and then fish will eat a whole bunch of them, so they'll have more heavy metals in their system per the amount of mass. But then eagles, you have to eat lots and lots of fish, so they have a massive amount of heavy metals in their system for the amount of mass they have, just because I've eaten so much stuff. It's accumulated through the food chain. And that's not all. Another thing that can happen is, as we're talking about hormones anyway, is that female contraceptive hormones in particular can cause feminization of aquatic organisms and reduce the sperm counts. So basically you're putting a lot of female hormones into the waters, and there are quite a few male animals will develop characteristics that are much more female, and they won't be able to reproduce properly. So female contraceptive hormones cause feminization of aquatic organisms and can reduce sperm counts. Okay, so that is a lot of information for a 6-mark question. They do want to see a lot of information for this question. You're just going to have to, for this question, I would put in more than what you would think 6 marks are, just in case. Just make sure everything that you put down is correct. Question 6. Figure 6.1 shows the changes in glucose concentration of the blood. Okay, as time goes on, the glucose concentration gets high, and then it goes low, then it goes higher, back to normal, then it'll go high, then low, then high. It never goes flat, exactly where it should be. A. Name the process that maintains blood glucose concentration within set limits. Okay, that is homeostasis. You could also say negative feedback, because negative feedback does the same thing. It's basically homeostasis. It's how homeostasis works. B. 1. Name the hormone that would be secreted in response to the increasing blood glucose concentration at A in figure 6.1. So when blood glucose levels go up, something needs to bring it down. That one will be insulin. We'll bring it down. To bring it back up, that will be glucagon. I always remember glucose is gone from the muscles. All right, that's glucagon. So at A, it is insulin. 2, name an organ that is responsible for the decrease in blood glucose concentration after B in figure 6.1. The organ that is responsible for the decrease in blood glucose concentration is here. The organ that does this, that is, can be 3 things. You can either say it's the liver, because the liver stores glucose as glycogen. Or you can say it's muscles, because the muscles increase respiration in response to insulin. And you could say it's the pancreas, because the pancreas produces the insulin. So all of those are responsible for decrease in blood glucose concentration, but only one. I'm going to say the liver. All right. 3, name the compound that is converted to glucose at C in figure 6.1. Okay, so here we have C. So the insulin has caused glucose to be stored as glycogen, and glycogen is converted to glucose by the glucagon. So it's glycogen that is converted. Glycogen. All right. C, describe the symptoms and the treatment of type 1 diabetes. Okay, so first of all, we'll go over the symptoms. There are lots and lots of symptoms. It's a 5-mark question. You don't have to put down every single one, but put down as many as you remember. Okay, symptoms. We have fatigue or tiredness. They'll be thirsty. They'll have increased urination. And that your urine contains glucose. And that's a test for diabetes. Urine contains glucose. Weight loss is a symptom. And we have nausea. They'll feel ill. They might vomit. They might have hunger. So they'll lose weight, but they'll be very hungry. And they could have blurred vision. They're not using their sugars as they should be using them. They could have glaucoma. Okay, I'm going to stop writing them down. You can't, the maximum of 3 marks from this section, so I'll just say some more. You could have behavioral changes, so that people might become confused. They may become faint. They can become unconscious or go into a coma. They can become very dizzy. They can have rapid breathing. They can have their wounds will heal slowly, and they can have poor circulation. So for symptoms, for this question, you won't get any more than 3 marks for the symptoms. So put down what you know. Maybe put down 4 just in case you're unsure, but but be sure that what you put down is correct. Okay, treatment. So the treatment for type 1 diabetes. Okay, insulin is injected when the glucose levels get too high. So insulin is injected when glucose levels get too high. And they figure out when they're too high by having regular blood glucose tests. This is determined by regular blood glucose tests, multiple times a day. And so that's how it's treated when it gets too high. Another way is that it can be controlled by making sure that the person with type 1 diabetes has regular meals and their diet is controlled. They don't have too much sugar or too little sugar. So it can also be controlled by regular meals and a controlled diet. All right, so that is the end of this question, and it is the end of this exam. I really hope that you found this useful and you'll do really, really well in your final exam. If you have found it useful, please press the like button, thumbs up. And if you haven't subscribed already, we'd really appreciate it if you did. And please leave any comments that you have for us in the discussion section below, as we really like to hear from you. And have a great day.